Hello students, let us continue with the next part of 14th chapter. So in the last part, we saw how plants respire. We learned about glycolysis and Krebs cycle. So after the TCA cycle, the following steps in the respiratory processes are to release and utilize the energy stored in NADH plus H plus and FADH2. So this is accomplished when they are oxidized through the electron transport system and the electrons are passed onto the oxygen, resulting in formation of water. So this is what we are going to study now. Electron transport system is a metabolic pathway present in the inner mitochondrial membrane through which the electron passes from one carrier to another. This is to release and utilize the energy stored in NADH, H plus and FADH2 which we got from the TCA cycle by oxidation. So the electrons are passed on to oxygen and we have a water molecule. Electrons from NADH are oxidized by an NADH dehydrogenase complex 1. Electrons are then transferred to ubiquinone located within the inner membrane. Ubiquinone also receives reducing equivalence by the FADH2, which is the complex 2, that is generated during oxidation of succinate in the citric acid cycle. So the reduced ubiquinone is then oxidized with the transfer of electrons to cytochrome C by cytochrome BC1 complex, which is the complex 3. Cytochrome C is a small protein attached to the outer surface of the inner membrane and it acts as a mobile carrier. So this mobile carrier carries the electrons between complex 3 and complex 4. So what is this complex 4? It is basically cytochrome C oxidase complex which has cytochrome A and A3 and two copper centers. When the electrons pass from one carrier to another by the complex 1 to 4, they are coupled with ATP synthase, that is complex 5, for the production of ATP. So complex 1 is NADH dehydrogenase, complex 2 has FADH2, 3 is cytochrome BC1, 4 is cytochrome C oxidase, and 5 has the ATP synthase. So in this way, the number of ATP molecules produced depends on the nature of the electron donor. Oxidation of one NADH will give 3 ATPs, whereas the oxidation of FADH2 will give 2 ATPs. So this is the overall electron transport chain where the electrons are transferred from complex 1 to 2 to 3 to 4 and eventually to water molecules. In aerobic respiration, the role of oxygen is limited to the end stage. But oxygen is vital because it drives the entire process by removing hydrogen from the system. So it acts as a final hydrogen acceptor. In respiration, energy of oxidation reduction is used for phosphorylation. So this process is called oxidative phosphorylation. Oxidative phosphorylation is different from photophosphorylation because in photophosphorylation, as the word suggests, light is used for the production of the gradient. The energy released during the electron transport system is used for the synthesis of ATP with the help of ATP synthase, which is complex 5. So ATP synthase has two components, the F1 headpiece, that is the peripheral membrane protein complex, and FO, which is the integral membrane protein complex. So these are the complexes F1 and FO. F1 is the site for ATP synthesis from ADP and inorganic phosphate. So ATP will join with phosphate molecule In this way, adenosine diphosphate will be converted into adenosine triphosphate with the help of the enzyme ATP synthase. The integral membrane protein complex forms a channel through which protons cross the inner membrane. So this is from where the 2H plus, the proton will cross this membrane. The passage of the proton is coupled to the catalytic site of F1 component for ATP production. So for each ATP produced, 2H plus passes through the complex from intermembrane space to the matrix down the electrochemical proton gradient. So if we look at the balance sheet of this entire respiratory process after glycolysis, Krebs cycle, and chemiosmosis, that is the electron transport chain, then what do we have? The net gain of ATP for each glucose molecule is calculated based on the following assumption. All the steps in glycolysis, GCase cycle, and electron transport chain occur sequentially and in correct order. The NADH synthesized in glycolysis is transferred into mitochondria and undergoes oxidative phosphorylation. The intermediates in the pathway are not used to synthesize other compounds. Only glucose is being respired. Other alternative substrates are not entered in the pathway at any stages. So based on all these conditions, we do the calculation of how many ATPs are produced. Such assumptions are not valid because all pathways work simultaneously and they do not take place one after another. Substrates enter the pathways and are withdrawn from it when the cell requires them. ATP is used when the cells require it. Enzymatic reactions are controlled by multiple means. So such calculations are used to appreciate the efficiency of living system in extraction and storage of energy. But we need to understand that each cell in our body works according to different situations. So during glycolysis, we have 2 ATPs which are directly made and 6 ATPs which we get from 2 molecules of NADH. During oxidative decarboxylation, we have 6 ATPs from 2 NADH. During TCA cycle, we have 18 ATPs from 6 NADH, 4 ATP from 2 FADH, 2 ATPs from 2 GTPs. So total, we've got 38 ATPs. 2 ATP molecules are spent for transporting the 2 NADH molecules formed during glycolysis to mitochondria because that is where the reaction has to occur. So the net gain is 36 ATPs. So as we know that aerobic respiration gives us more energy compared to anaerobic respiration. Why is that? This is because in fermentation there is partial breakdown of glucose, whereas here there is complete breakdown of glucose to carbon dioxide and water. Here, as I was telling you, there are 36 ATPs, here there are only 2 ATPs. NADH is oxidized to NAD plus very slowly here, whereas here the oxidation is very vigorous. Now let us learn about the amphibolic pathway. Glucose is the favored substrate for respiration. So all carbohydrates are first converted into glucose for respiration. Other substrates are also respired. Fats are broken down into fatty acids and glycerols. Fatty acids are degraded to acetyl-CoA and they enter the pathway. Glycerol is converted into PGAL and then it enters the pathway. Proteins are degraded by proteases to amino acids. Each amino acid, after deamination, that is the removal of amino group, enters the pathway at some stage in the Krebs cycle or as pyruvate or as acetyl-CoA. So the general assumption is that respiratory pathway is a catabolic pathway because energy is released and things are broken down out here. But here, as we see, both anabolism, that is synthesis, as well as catabolism, that is breakdown, is happening. So we call it amphibolic pathway. For example, fatty acids are broken down to acetyl-CoA before entering the pathway, but when the organism needs to synthesize fatty acid, acetyl-CoA withdraw from the respiratory pathway. Similarly, during breakdown and synthesis of protein, respiratory intermediates are involved, so we have to call it amphibolic pathway. Now let us learn what is respiratory quotient. It is the volume of CO2 evolved to the volume of oxygen consumed. So this depends on the type of respiratory substrate. For carbohydrates, it is 1 because equal amount of CO2 and O2 are evolved and consumed respectively. So 6 CO2 upon 6 O2 will give you 1. For fats, it can be less than 1 because calculation for fatty acids can be like this. 102 CO2 upon 145 oxygen gives zero point 0.7, which is less than 1. For proteins, it is zero point 0.9. So in the living organism, respiratory substances are often more than 1. Pure proteins or fats are never used as respiratory substrates. I hope you understood this chapter properly. Thank you.